The Permian period lasted from 299 to 251 million years ago, and it was the last period of the Paleozoic Era. This distinction between the Paleozoic and the Mesozoic is made at the end of the Permian in recognition of the largest mass extinction recorded in the history of life on Earth. It affected many groups of organisms in many different environments, but it affected marine communities by far the most, causing the extinction of most of the marine invertebrates of the time. Some groups survived the Permian mass extinction in greatly diminished numbers, but they never again reached the ecological dominance they once had, clearing the way for another group of sea life. On land, a relatively smaller extinction of diapsids and synapsids cleared the way for other forms to dominate and led to what has been called the Age of Dinosaurs. Also, the great forests of fern-like plants shifted to gymnosperms, plants with their offspring enclosed within seeds. Modern conifers, the most familiar gymnosperms of today, first appeared in the fossil record of the Permian. The Permian was a time of great changes, and life on Earth was never the same again. The global geography during the Permian period was characterized by vast expanses of land and water. By the start of the Permian, the movement of Earth's tectonic plates has consolidated much of the planet's landmass into a supercontinent called Pangaea. This landmass included most of today's continents, largely intact, except for Asia, which was fragmented at the time. Pangaea extended from the northern to the southern pole, while the majority of Earth's remaining surface was covered by a vast ocean called Panthalassa. To the east of Pangaea, there was a smaller sea known as Tethys. The interior regions of Pangaea were likely dry, with significant seasonal fluctuations due to the absence of nearby bodies of water to moderate the climate. Only certain parts of this vast interior received consistent rainfall throughout the year. Little is known about the Panthallassic Ocean itself, but the evidence suggests that Earth's climate shifted during the Permian, with a decrease in glaciation as continental interiors became drier. The early Permian is believed to have been relatively cool, following the glaciations of the late Carboniferous period. However, as the period progressed, there was a gradual warming trend. By the late Permian, global temperatures had risen, leading to the melting of polar ice caps and a reduction in glacial activity. This warming trend contributed to the drying out of continental interiors and the expansion of arid environments. The Permian climate was also influenced by changes in atmospheric composition. There is evidence to suggest that levels of carbon dioxide increased during this period, possibly due to volcanic activity and the release of gases from the Earth's interior. Higher CO2 levels would have contributed to the greenhouse effect, further warming the planet. In addition, oxygen levels in the atmosphere were relatively high during the early Permian, but began to decline towards the end of the period. This decline in oxygen may have had significant impacts on the biosphere, particularly for large terrestrial animals that were adapted to higher oxygen levels. The late Permian period was marked by a series of climatic and environmental changes that culminated in the end Permian mass extinction, also known as the Permian-Triassic extinction event. This event, which occurred around 252 million years ago, is considered the most severe extinction event in Earth's history, with an estimated 90% of marine species and 70% of terrestrial vertebrate species going extinct. The causes of this extinction are still debated, but it is likely that a combination of factors, including volcanic activity, climate change, ocean acidification, and anoxia, or a lack of oxygen, played a role. Volcanic activity, particularly the massive eruptions in the Siberian Traps, is believed to have been a significant driver of the late Permian climate crisis. These eruptions released enormous amounts of volcanic gases, including CO2 and sulfur dioxide, into the atmosphere, leading to rapid climate change. The release of CO2 would have contributed to global warming, while SO2 could have caused acid rain and further disrupted ecosystems. The Permian was a time of specialization for marine fauna, with major diversifications of ammonoids, brachiopods, and bryozoans. Insects, amphibians, and therapsids, the precursors of mammals, flourished during this time. Reptiles began to flourish in water and on land. During the middle of the Permian, there was a major transition in vegetation, resulting in a dramatic loss of coal swamps and amphibian habitats. Conifers and other xerophytic seed plants, better adapted to the changing climate conditions and drier environments of the Permian, appeared and spread. Competition in the dry climate belts led to more diversity and variability in seed plants. Continuing evolution of arthropod herbivores and the first appearance of vertebrate herbivores encouraged plant evolution in new directions. Insects were the most important herbivores of the early Permian. Non-metamorphosing insects related to grasshoppers and cockroaches from the Pennsylvanian were joined by the first known insect groups to undergo metamorphosis. These new insect types, such as beetles, gained the advantage of the larva and adults utilizing different ecological resources, profoundly affecting insect-plant and insect-predator interactions and coevolution. With these new forms, insects reached their highest diversity of high-level forms of all time. Most major groups of terrestrial vertebrates were present in the Permian. A wide variety of amphibians lived on land and in fresh water, including the giant carnivorous Eryops, as well as large herbivores. The two major amniote vertebrate groups, synapsids, mammal-like reptiles, and diapsids, true reptiles, had diversified into many groups, including some of the first large vertebrate herbivores. While reptilian carnivores radiated to many forms and niches throughout the Permian, the invention of herbivory was the important and complex vertebrate adaptation. Plant material not only requires special mechanical adaptations to break down vegetable material, it is also largely indigestible, requiring specialized symbiotic relationships with fermentative bacteria. The terrestrial fossil record of the Permian is patchy and temporarily discontinuous. Early Permian records are dominated by equatorial Europe and North America, while those of the Middle and Late Permian are dominated by temperate Karoo supergroup sediments of South Africa and the Ural region of European Russia. Early Permian terrestrial faunas of North America and Europe were dominated by primitive pelycosaur synapsids, including the herbivorous Edaphosaurids and carnivorous Sphenacodontids, Diadectes, and amphibians. Early Permian reptiles, such as Cleistahinids, were mostly small insectivores. Dimetrodon is an extinct mammal-like animal that lived during the early Permian period, around 295 to 272 million years ago. Several ancient animal fossils have been discovered in the southwestern part of the US. Although it is commonly mistaken for a dinosaur, the Dimetrodon is older than the earliest dinosaurs by about 40 million years. Dimetrodon was an apex predator, known for the prominent spine sail on its back. The most prominent feature of Dimetrodon was undoubtedly its large sail. The sail projects outwards from its vertebrae and forms by extended neural spines entwined by a membrane of several blood vessels. Scientists believe that the iconic sail is mainly for temperature regulation. Other researchers doubt this is true, considering the Sphenacodon, a closer relative of Dimetrodon that also existed during the early Permian, had no sail. The first Dimetrodon fossils were discovered in the 1800s, with credit for the earliest finds going to Donald MacLeod, who lived in the British colony of Prince Edward Island. In 1845, MacLeod unearthed a maxilla which he later sold to Canadian geologist John William Johnson. In 1854, Johnson described this fossil as the mandible of Bathygnathus borealis, a carnivore related to Secodontosaurus. However, in 2015, modern paleontologists reclassified it as belonging to Dimetrodon, making it the first Dimetrodon jaw with zyphodont teeth discovered in Canada. Most Dimetrodon fossils have been found in a geological deposit known as the Red Beds in Texas and Oklahoma. Edward Drinker Cope was the first to publish an official description of this animal. He obtained the fossil for his description, along with those of many other Permian tetrapods, from several collectors who had been exploring these famous red beds in Texas. Dimetrodon had a tall, laterally compressed skull, featuring incisor-like front teeth and a large canine tooth, followed by a row of smaller teeth. The structure of its teeth and the large openings behind its eyes, known as temporal fenestrae, have been crucial for paleontologists in identifying and studying the species. The average Dimetrodon size ranged between 1.8 to 3.4 meters, or 6 and 11 feet, with an estimated weight of 27 to 181 kilograms, or 60 to 400 pounds. However, the most prominent member of this genus, Dimetrodon angelensis, was slightly larger, reaching about 4.6 meters or 15 feet in length, and weighing around 250 kilograms, or 550 pounds. Dimetrodon was sexually dimorphic, meaning that males were larger than females. Dimetrodon was a carnivore, primarily feeding on fish and other amphibians. Scientists inferred this diet by analyzing and comparing the teeth of 3 different Dimetrodon species. The variations in tooth size and shape suggest that Dimetrodon evolved over time to hunt larger prey. As its regular prey grew larger in response to competition from other large carnivores, Dimetrodon also had to increase in size to keep up with the changing ecosystem. Its sharp canines at the front of its snout were ideal for piercing and holding on to freshly killed prey, while the teeth at the back were specialized for shearing bones and grinding tough muscles. Dimetrodon went extinct by the end of the Permian period due to a catastrophic event known as the Great Dying, or the Permian extinction. This disaster was caused by significant global warming, which led to widespread changes in the oceans and other ecosystems. The Great Dying resulted in the extinction of over 90% of dinosaur species and 97% of all life on Earth, affecting both land and marine species. Lystrosaurus is one of the few land animals that survived the Permian-Triassic extinction, one of the most significant extinction events in geologic history. This pig-sized reptile lived over 250 million years ago and, at one point during the early Triassic, accounted for 95% of land vertebrates. The discovery of Lystrosaurus fossils has been crucial in providing evidence for continental drift and the existence of ancient land bridges. Lystrosaurus, an extinct genus of Dicynodont therapsids that roamed the Earth during the early Triassic and late Permian periods. The name Lystrosaurus comes from the Greek words lystron sauros, meaning shovel lizard, and it is easy to see why. Lystrosaurus measured between 0.9 and 2.4 meters, or 3 to 8 feet long, and weighed around 90 kilograms or 200 pounds, roughly the size of an average pig. Its appearance was somewhat pig-like too, with a short snout, though it only had 2 tusk-like upper canines instead of a full set of teeth. These tusks were deeply set in the upper jaw, and wear patterns suggest they were used for digging and rooting out vegetation. It also had horny beaks, similar to those of turtles, perfect for shearing off plants. One interesting feature of Lystrosaurus was its jaw, which had a unique joint that moved in an upward and backward motion for shearing instead of the typical up and down or sideways movement. The lower jaw could also slide forward and backward, making it easier for the animal to move vegetation into its mouth. Its nostrils were located high on its curved snout, an adaptation that may have allowed it to feed in shallow waters or marshy environments. In terms of posture, Lystrosaurus had a sprawling stance with a short tail and 5 toes on each foot. The front legs were larger and stronger than the rear ones, suggesting that Lystrosaurus was a powerful digger and burrower. Its legs were slightly splayed out to the sides, similar to how alligators walk today. The 5 sacral vertebrae were massive, but not fused to each other or the pelvis, giving Lystrosaurus a rigid back. Bone histology indicates that Lystrosaurus was a fast-growing animal, and mummified specimens from the Karoo Basin show that it had dimpled, leathery, and hairless skin. Lystrosaurus inhabited various ecosystems across a wide geographical range, leading to adaptations that allowed it to consume different types of plants. Many species of Lystrosaurus fed on Dicroidium, an extinct genus of fork-leaved ferns that thrived during the Permian and Triassic periods. Larger species, such as Lystrosaurus macaigi, likely evolved to rely on bigger plants of the time, like the Glossopteris flora. Unfortunately, this plant did not survive the Permian extinction. Dr. Elias Root Beadle, a Philadelphia missionary and passionate fossil collector, discovered the first Lystrosaurus skull. He reached out to the renowned paleontologist Othniel Charles Marsh about his find, but never received a response. Instead, Edward Drinker Cope, Marsh's major rival, took an interest in the discovery and named Lystrosaurus in 1870. Most Lystrosaurus fossils have been unearthed in the Balfour and Katberg formations of the Karoo Basin in South Africa. While the initial estimate of species discovered between the 1930s and 1970s was 23, further research in the 1990s reduced the number to just 6 recognized species. The absence of predators and herbivore competition for the available food allowed Lystrosaurus to thrive without restraint, so much so that for a while 95% of land vertebrates were Lystrosaurus, becoming the most common group of terrestrial vertebrates during the early Triassic. Inostrancevia was a Gorgonopsian therapsid that lived around 259 to 251 million years ago during the late Permian period. This unique creature roamed the prehistoric landscapes of what is now European Russia and South Africa. Inostrancevia is believed to have been one of the largest of its kind and an apex predator of its time. One of the most striking features of Inostrancevia is its large canines, which were perfect for taking down prey with efficiency. It was fully terrestrial, roaming the cold desert regions of ancient Russia. With a large skull to accommodate its impressive teeth, Inostrancevia had a relatively long and robust neck to support its massive head as it moved through its environment. Inostrancevia was a highly robust non-mammalian therapsid, especially notable for its strong limbs and broad skull, with a raised, elongated snout. Within the genus, Inostrancevia latifrons had a lower and wider snout compared to Inostrancevia alexandri, which had a larger skull overall. The eye sockets of Inostrancevia were relatively small, and its powerful jaws featured strong, finely serrated teeth, adapted for gripping and tearing flesh, with the upper canines reaching lengths of 12 to 15 centimeters or 4.7 to 5.9 inches, the largest known canines among non-mammalian therapsids. Its long neck, probably consisting of 7 cervical vertebrae, supported its large head. The hands and feet each had 5 digits, with the 5th digits not attached to the carpus or tarsus, but extending from the ulna and the heel bone. The pelvis retained a reptilian structure, and its femur had a distinctive curved S shape, longer than the humerus, where the tail was relatively short. Inostrancevia was one of the largest Gorgonopsians, with Inostrancevia alexandri specimens reaching about 3 meters or 9.8 feet in length, and skulls measuring approximately 50 centimeters or 20 inches long. Inostrancevia latifrons might have been even larger, though this remains uncertain due to incomplete fossils. Based on a 60 centimeter or 23.6 inch skull, scientists estimate that this beast could have reached a total length of 3.5 meters or 11.5 feet and weighed around 300 kilograms or 661 pounds. During the late Permian, the region of Russia inhabited by Inostrancevia was a cold desert characterized by hot, dry summers and cold, dry winters with minimal snowfall, while rivers and lakes were present in the habitat. At that time, European Russia was dominated by plants from the Peltospermaceae family, with ginkgophytes and conifers being quite common, while ferns and sphenophytes were relatively rare. Although many portrayals depict Inostrancevia swimming while pursuing prey, it was likely a fully terrestrial carnivore adapted to the lands. Its limbs were probably positioned nearly vertically rather than horizontally, although it could not have moved them too far forward. It is widely believed that Gorgonopsians, including Inostrancevia, were nocturnal and adapted to low-light conditions, though only Clelandina has been confirmed as strictly diurnal. While there is no specific evidence regarding the behavioral patterns of Inostrancevia, it is possible that it hunted primarily at night. Gorgonopsians generally had strong bite forces, but those with less robust skulls and longer canines, such as Inostrancevia and Scutosaurus, likely had weaker bites and relied on their canines to slash prey. For instance, a similar Gorgonopsian, Rubigia, had a bite force of approximately 715 newtons, suggesting that Inostrancevia had a comparable bite force. Inostrancevia may have used a bite and retreat hunting strategy, ambushing its prey with a powerful bite and then following as the prey attempted to escape, ultimately delivering a final bite. The reproductive behavior and life cycle of Inostrancevia and other therapsids remains poorly understood. While some sources speculate that Inostrancevia laid eggs, there is no supporting evidence. Comparisons with mammals suggest that therapsids might have had anal and urogenital openings, mammal-like facial musculature for suckling, and possibly mammary glands, leading to theories of viviparity. However, these aspects remain unconfirmed for Inostrancevia, and details such as the gestation period, neonatal development, and parental care practices are unknown. Anteosaurus is a genus of large mammal-like reptiles that lived during the Permian period, approximately 266 to 260 million years ago in what is now South Africa. Although it had a semi-aquatic lifestyle similar to modern crocodiles, Anteosaurus is not related to crocodiles. This genus went extinct during the Middle to Late Permian, leaving no living descendants. Anteosaurus belonged to the synapsids, an ancient group that includes mammals and their close relatives, such as sauropods. The name Anteosaurus means early reptile or Antaeus reptile, referencing a figure from Greek mythology. It was likely the largest non-mammalian carnivore in the synapsid family, weighing between 500 and 590 kilograms, 1,100 and 1,300 pounds, and measuring about 5 to 6 meters or 16 to 19 feet in length. A prominent feature of Anteosaurus is its distinctive dentition. The genus had large teeth on the roof of its mouth, clustered near the outer tooth row, with a dental arrangement including anterior, canine, and cheek teeth. The anterior teeth had a ledge, a trait shared with other Dinocephalians, while the large, prominent canine teeth and 10 smaller cheek teeth were characteristic of this genus. The thick skull of Anteosaurus is believed to be an adaptation for headbutting, likely used in territorial disputes or mating behavior. This suggests that Anteosaurus was as active on land as it was in water. Initially thought to be sluggish, crocodilian-like predators that relied on stealth and could drag prey into the water, more recent studies suggest that this view is inaccurate. Evidence indicates that Anteosaurus was a fast and agile predator with well-developed senses, including a large inner ear and a significant portion of the brain dedicated to eye movement, making it an effective apex predator capable of swift hunting and rapid strikes. Anteosaurus lived about 30 million years before the first dinosaurs appeared. The skull of a juvenile Anteosaurus was discovered in 1921 in the Eastern Cape province of South Africa. While numerous dinocephalian skull fragments have been found in South Africa and Russia, Anteosaurus magnificus is the only species of this genus identified to date and is the sole member of the Anteosauridae family found in the South African temperate zone. During the Middle to Late Permian, Anteosaurus and other synapsids were among the dominant terrestrial animals. However, they were likely wiped out by the Permian Triassic mass extinction event about 250 million years ago. The reproductive behavior of Anteosaurus is not well documented. While some theories suggest egg-laying, there is no direct evidence. Comparisons with mammals suggest it might have been viviparous, but this remains speculative. The study of Anteosaurus has provided valuable information on the evolution and diversity of early synapsids. The robust nature of its skull and teeth has contributed to understanding the adaptations and lifestyle of these prehistoric creatures. Scutosaurus, shield lizard, is an extinct genus of Pareiasaur parareptiles. Its genus name refers to large plates of armor scattered across its body. It was a large anapsid reptile that, unlike most reptiles, held its legs underneath its body to support its great weight. Fossils have been found in the Sokolki assemblage zone of the Malokinelskaya Formation in European Russia, close to the Ural Mountains, dating to the late Permian period between 264 and 252 billion years ago. Scutosaurus was a large Pareiasaur, weighing up to 1,160 kilograms or 2,560 pounds, comparable in size to a modern-day black rhinoceros. Its body length ranged from about 2.5 to 3 meters, or 8 to 9 feet. The entire body was covered with osteoderms, bony plates that were separate rather than forming a continuous armor, but were closely packed together. Similar to other Pareiasaurs, Scutosaurus had a barrel-shaped ribcage and massive limbs. It featured a short, stout body with a very short tail. Scutosaurus karpinskii had an exceptionally thick skull adorned with unusual bony bumps, giving it an appearance reminiscent of early horned dinosaurs like Triceratops. Its broad torso supported a large digestive system. The snout of Scutosaurus was broad, equipped with rows of blade-like heterodont teeth. The upper jaw had 18 teeth, while the lower jaw had 16. Additionally, this species had a small bony projection, or tubercle, behind the skull, a unique feature not found in other Pareiasaurs. All known fossils of Scutosaurus date back to the Lopingian epoch of the Upper Permian period, approximately 259 million years ago. Compared to other members of the Pareiasaur family, Scutosaurus evolved to achieve notably larger sizes. In addition to its increased size, Scutosaurus exhibits another significant evolutionary adaptation: the absence of the clithrum in its shoulder blade. The clithrum, a membrane bone first seen in primitive fish and present in all their descendants, including early Pareiasaurs, was lost in later Pareiasaurs such as Scutosaurus. The loss of this structure likely contributed to the development of a more flexible and freely movable neck in subsequent animals. Despite its imposing build, Scutosaurus was a herbivore, living in semi-arid environments where it either traveled long distances for food or stayed near floodplains with abundant vegetation. Its sharp, flat teeth were adapted for chewing leaves and young branches, and it used gastroliths or stomach stones to aid digestion. Due to its large size, Scutosaurus needed a constant food supply and could not survive without regular nourishment. Parareptiles like Scutosaurus had relatively short legs, which meant they could only run quickly for short bursts. This made them easy targets for larger predators. To compensate, they had thick skeletons and strong muscles, especially in their necks, which made them tough to kill despite their slow speed. Scutosaurus also had bony plates under its skin that provided additional armor. However, even with these defenses, larger predators like Inostrancevia, with its enormous saber-like teeth, still preyed on them. Estemmenosuchus is a genus of extinct omnivorous therapsids that roamed the Earth during the late Permian period, roughly 260 million years ago. This genus, a member of the Gorgonopsians, represents a significant chapter in the history of prehistoric predators. Known for its impressive physical characteristics and evolutionary significance, Estemmenosuchus provides valuable insights into the ancient ecosystems and the evolutionary processes that shaped the terrestrial vertebrate lineage. The genus Estemmenosuchus was first described in the early 20th century from fossils discovered in the Karoo Basin of South Africa and parts of Russia. The name Estemmenosuchus means crowned crocodile, a reference to the distinctive cranial features of the species that resemble a crown-like arrangement of bony projections. These projections and the overall robust build of Estemmenosuchus make it one of the most remarkable Gorgonopsians known to paleontology. Estemmenosuchus was a large and formidable predator, measuring approximately 3 to 4 meters or 10 to 13 feet in length, and weighing around 600 kilograms or 1,300 pounds. Its size and body structure positioned it as a top predator in its ecosystem. The skull was broad and flat, adorned with several bony ridges and protrusions that created a crown-like appearance. These cranial features were likely used for display purposes, combat, or both, giving Estemmenosuchus an imposing and a distinctive look. The skull of Estemmenosuchus was equipped with large, saber-like canine teeth, which were designed for slicing through flesh and crushing bone. These canines were complemented by smaller, serrated post-canine teeth that aided in shearing and grinding. The jaw structure of Estemmenosuchus was robust and capable of delivering a powerful bite, making it an effective predator of its time. In addition to its impressive dental arsenal, Estemmenosuchus had a sturdy, heavily built body. Its limbs were robust, supporting its large frame and enabling it to move with a combination of speed and power. During the late Permian period, the environment where Estemmenosuchus lived was characterized by a mix of semi-arid and lush, Floodplain ecosystems. The climate of this period was marked by significant fluctuations, with hot, dry phases interspersed with wetter conditions. Fossil evidence suggests that Estemmenosuchus inhabited regions that are now part of modern-day South Africa and Russia. The genus Estemmenosuchus is of considerable importance in the study of early therapsids, a group of synapsids from which mammals eventually evolved. As a Gorgonopsian, Estemmenosuchus represents an early branch of therapsids that exhibited characteristics leading towards more advanced forms. The evolution of Estemmenosuchus and its relatives is crucial for understanding the transition from early reptiles to more specialized therapsids. The adaptation of large canines and a robust body structure reflects the diversification of predatory strategies during this time. The first fossils of Estemmenosuchus were uncovered in the early 20th century, and subsequent discoveries have continued to provide valuable insights into its biology. The analysis of these fossils has revealed much about its predatory nature, physical adaptations, and the environmental conditions in which it lived. Research in Estemmenosuchus has shed light on the evolutionary processes that shaped the development of therapsids and their eventual transition to mammalian forms. The study of its fossils has provided valuable information about the diversity of predatory strategies employed by early reptiles and the evolutionary path leading to modern mammals. Diplocaulus is an extinct early amphibian that lived 270 million years ago during the Permian period. It was first discovered in 1878 in Texas and was given its name by Edward Drinker Cope in 1877. Its name means double caul because it has a very distinctive head that looks like a boomerang. If you saw pictures of Diplocaulus without any context, you might think it was a product of someone's imagination. In reality, Diplocaulus was a genuine amphibian with a distinctive, unusual head shape. This unique feature likely served as a defense mechanism, making it less appealing to predators. Its oddly shaped head would have made it challenging for many prehistoric aquatic creatures to swallow, leading them to avoid it in favor of easier prey. Diplocaulus had a stocky, salamander-like body, but was relatively large, reaching up to 1 meter or 3.3 feet in length. Although a complete tail is unknown for the genus, a nearly complete articulated skeleton described in 1917 preserved a row of tail vertebrae near the head. This was construed as circumstantial evidence for a long, thin tail capable of reaching the head if the animal was curled up. Most studies since this discovery have argued that anguilliform or eel-like tail movements was the main force of locomotion utilized by Diplocaulus and its relatives. Other scientists believe that its head may have been used as a hydrofoil. This would have made it possible for it to skim on top of a body of water. If that's true, then the shape of its head could have also been used as a way for this animal to steer itself. This would have allowed it to traverse streams, rivers, and lakes with ease. The unique shape of the skull also indicates that Diplocaulus had a modified feeding mechanism. Its dentition was adapted for a diet that included small invertebrates, which it likely captured in aquatic environments. The teeth were small and conical, suited for grasping rather than chewing. Diplocaulus is thought to have primarily lived in aquatic environments, but could have ventured onto land occasionally. Its body structure suggests that it was a good swimmer, with a streamlined body and strong limbs. However, its relatively short legs and elongated body might have limited its terrestrial mobility compared to fully aquatic or fully terrestrial animals. During the time Diplocaulus lived, the climate was generally warm and humid, with extensive swampy areas and lush vegetation. The environment was characterized by large, slow-moving rivers and abundant plant life, providing a rich habitat for amphibians. Diplocaulus likely coexisted with other prehistoric amphibians, early reptiles, and various aquatic invertebrates. Diplocaulus belonged to the Lepospondyli, a group of early amphibians characterized by their unique vertebral structure. Lepospondylis were diverse and adapted to a variety of ecological niches, but they eventually became extinct by the end of the Permian period. The exact reasons for their extinction are not fully understood, but are likely related to the dramatic environmental changes and the rise of new vertebrate groups. Titanophoneus, or Titanic murderer, is an extinct genus of carnivorous dinocephalian therapsids from the Middle Permian period. It belongs to the family Anteosauridae, with the Titanophoneus potens being the type species. Fossils of Titanophoneus have been uncovered in Ishivo, Russia. The skull of an adult Titanophoneus could reach up to 80 centimeters, about 31.5 inches in length, featuring a robust, elongated snout. This therapsid had a long tail and short limbs, which are indicative of its primitive nature compared to more advanced theropods, like Inostrancevia. The limb structure and bone density of Titanophoneus suggests it maintained a sprawling posture. The temporal fenestra of Titanophoneus is more developed than in earlier forms such as Estemmenosuchus, but less advanced than in Inostrancevia. Its dentition includes 12 large incisors on the palate, 2 prominent canines, and various smaller teeth towards the back. The lower palate has a similar arrangement to the upper but lacks the canine teeth. The overall appearance of Titanophoneus is reminiscent of the sphenacodontid pelycosaurs, such as Dimetrodon, indicating its position within the broader evolutionary context of synapsids. While not directly related to these earlier pelycosaurs, Titanophoneus shares some morphological features with them, highlighting its ancient lineage within the therapsid clade. Titanophoneus belonged to the group Therapsida, which includes both non-mammalian and early mammalian forms. Most early therapsids are thought to have been oviparous or egg-laying, like their synapsid ancestors. This assumption is based on the reproductive strategies observed in modern relatives and early mammal-like reptiles. However, as therapsids evolved, some groups, especially closer to the origin of mammals, exhibited viviparity. Titanophoneus lived during the Middle Permian, which is before the known evolution of viviparous therapsids. Thus, it is more likely that Titanophoneus laid eggs. Evidence for parental care in early therapsids is also limited. However, many modern reptiles and amphibians exhibit varying degrees of parental care, and it is possible that Titanophoneus may have had some form of protection or care for its young. This care could have included providing a safe environment for the hatchlings until they were capable of fending for themselves. Gorgonops is an extinct genus of therapsid that lived approximately 260 to 254 million years ago during the late Permian period. As a notable representative of the suborder Gorgonopsia, Gorgonops was among the dominant predators of its time, with the largest species reaching lengths of 3 meters. Gorgonops was a medium-sized therapsid, measuring between 1.2 to 2 meters or 4 to 6.6 feet from snout to tail, depending on the species. The skull length ranged from 22 to 35 centimeters or 8.7 to 13.8 inches. This genus possessed a robust, deep skull with a triangular profile when viewed from above. One of its most striking features was the pair of oversized canine teeth, which could reach lengths of up to 12 centimeters or 4.7 inches. These formidable teeth nearly extended beyond the lower jaw. Gorgonops was a key predator in its ecosystem, primarily due to its powerful dentition. The enlarged canines were adept at penetrating the tough hides of contemporary herbivores, such as Pareiasaurus, like Brachiosaurus. The structure of the lower jaw was adapted to protect these large teeth from damage. The anterior portion of the jaw was thicker than the posterior, a feature reminiscent of the bone flanges seen in saber-toothed cats. This adaptation likely provided additional support and protection for the canines during aggressive encounters. The limbs of Gorgonops were positioned directly beneath the body, a trait that differed from the sprawling limb postures seen in many prey species of the time. This arrangement allowed for more efficient locomotion and greater speed, making Gorgonops a more effective hunter. The more upright limb position also facilitated rapid bursts of speed, enhancing its predatory capabilities. Recent studies of Gorgonops have emphasized its role in the late Permian ecosystems and its evolutionary importance. The genus is considered a key representative of the Gorgonopsia suborder, which includes some of the most advanced and largest therapsids of the period. Fossil evidence, including well-preserved skulls and limb bones, has contributed to our understanding of its anatomy, behavior, and ecological role. While direct evidence of Gorgonops' reproductive behavior is limited, it is inferred from comparisons with other therapsids and living analogues. Gorgonops likely exhibited reproductive strategies similar to other large predators of the time, possibly laying eggs or giving live birth, though the exact mode of reproduction remains uncertain. Behavioral insights into Gorgonops include its probable solitary or territorial nature, given its size and predatory habits. Moschops, an intriguing genus of dinocephalian synapsids, thrived approximately 265 to 260 million years ago during the Capetanian age of the Permian period. As members of the dinocephalian suborder, these creatures were distinguished by their large, heavily built bodies and mammal-like postures. Moschops exhibited a robust and distinctive anatomy. They had relatively small heads with high, short snouts, broad orbits, and strong jaw muscles. The creature's body was supported by strong forelimbs and hindlimbs, with a relatively wide pelvis. Notably, the shoulder blades were positioned significantly higher than the pelvis, and the limbs were adapted for a more mammal-like movement, as opposed to the sprawling posture seen in many reptiles of the time. The anatomical arrangement allowed Moschops to move with greater efficiency and speed. In terms of size, Moschops reached approximately 2.7 meters or 8.9 feet in length and weighed around 129 kilograms or 284 pounds, with some specimens recorded at up to 327.4 kilograms or 722 pounds. When compared to their closest relatives in the Tapinocephalidae family, Moschops was relatively lighter. For example, Rheabecosaurus, which measured about 2.5 meters or 8.2 feet long, weighed approximately 500 kilograms or 1,100 pounds. Similarly, Keratocephalus, also around 2.4 to 3 meters or 8.2 to 9.8 feet in length, reached weights between 700 and 1,000 kilograms. Tapinocephalus was even larger, reaching weights of 1.2 to 2 tons, 3,300 to 4,400 pounds, while maintaining a length of about 3 meters or 9.8 feet. One of the most remarkable features of Moschops was its thick skull, which housed a surprisingly small brain, approximately the size of a chicken egg. Despite its seemingly rudimentary brain, Moschops demonstrated effective survival and reproduction strategies, the skull's robust structure suggesting that Moschops may have engaged in headbutting behavior, likely for mating or territorial disputes. This hypothesis is supported by anatomical features indicating a capacity for combative interactions. Moschops inhabited a semi-arid environment, which required it to either travel significant distances to find food or stay close to floodplains and riverbanks where vegetation was more abundant. Its diet primarily consisted of tough vegetation, such as cycad stems, which was not particularly nutrient-rich. As a result, Moschops likely spent substantial time foraging to meet its nutritional needs. There has been some debate about whether Moschops was fully terrestrial or semi-aquatic. While some members of the Dinocephalian family are considered aquatic or semi-aquatic, Moschops' physical adaptations suggest it was primarily terrestrial. The mammal-like characteristics of its movement and behavior would have supported a fully terrestrial lifestyle. Its exact evolutionary lineage remains unclear. It is believed that Dinocephalians evolved during the Rhodian or Kungurian stages of the Permian period, possibly following the extinction of pelycosaurs due to competition. The discovery of Moschops fossils provides valuable insights into the evolution and diversity of early synapsids. The first Moschops fossils were discovered in the early 1900s in South Africa. These fossils were subsequently sent to the American Museum of Natural History in New York City, where they were described in 1906. Fossil records suggest the existence of two Moschops species, Moschops capensis and Moschops cuopensis. Recent studies focus on understanding the connections between therapsids and modern mammals. Research includes examining whether therapsids, including Moschops, were viviparous and identifying features such as mammary glands, anal and urogenital openings, and specialized facial musculature for suckling. Although evidence for these traits in Moschops remains inconclusive, studies of similar therapsids provide a broader context for understanding the evolutionary transition from early synapsids to mammals. Sphenacodon is an extinct genus of synapsid that lived from about 300 to about 280 million years ago during the late Carboniferous and early Permian periods. Like the closely related Dimetrodon, Sphenacodon was a carnivorous member of the Eupellicosauria family Sphenacodontidae. However, Sphenacodon had a low crest along its back, formed from blade-like bones on its vertebrae or neural spines, instead of the tall dorsal sail found in Dimetrodon. Fossils of Sphenacodon are known from the New Mexico and the Utah-Arizona border region in North America. Researchers currently recognize two species, Sphenacodon ferox, the type species, and Sphenacodon ferocior. Sphenacodon ferocior can be up to 40% larger in overall size, at about 3 meters or 9.8 feet long, compared to Sphenacodon ferox at about 2 meters or 6.6 feet. In addition, the dorsal spines in Sphenacodon ferocior are proportionately 45% taller than in Sphenacodon ferox. The recent discovery of a nearly complete skull of Sphenacodon ferox has helped clarify other distinctions between the two species, including the number of teeth in certain parts of the jaws and the size of the indented notch between the maxillary and premaxillary bones in the upper jaw. The two species occur together in some formations, but Sphenacodon ferox apparently survived later into the early Permian. Sphenacodon and Dimetrodon, two prominent genera of early synapsids, were typically found in distinct geographical regions, separated by the ancient Hueco Seaway, which divided equatorial Pangaea during the early Permian. This seaway covered much of southern New Mexico and parts of West Texas. Sphenacodon fossils have been discovered primarily in the western regions, including New Mexico, Arizona, and Utah. In contrast, Dimetrodon was predominantly found in eastern regions such as Texas and Oklahoma, which were characterized by deltaic environments. Notably, the species Dimetrodon occidentalis has also been identified in New Mexico. In their respective regions, both genera would have been apex predators, preying on a variety of animals including amphibians, Diadectes, and early synapsids and diapsids. Sphenacodon appears to have become extinct around 280 million years ago during the Wolf-Campian stage, whereas Dimetrodon persisted until approximately 270 million years ago. These large sphenocodontid predators were eventually succeeded by therapsids, a group of synapsids that includes the direct ancestors of mammals. Both Sphenacodon and Dimetrodon have traditionally been depicted with their limbs splayed outward at 90 degrees from their bodies, resembling a push-up stance with tails and bellies dragging on the ground, similar to modern lizards and crocodiles. This sprawling posture is reflected in museum reconstructions. However, fossil trackways, known as Dimetropus, show these animals walking with limbs held closer to the body in a more upright gait, without tail or belly drag marks. This suggests that their true anatomy and locomotion might not be fully understood. Some narrow Dimetrodon tracks found in New Mexico could match the smaller size of Sphenacodon, but might also belong to a smaller Dimetrodon. Details about the reproductive behavior of Sphenacodon are limited, but like other early synapsids, it is believed to have laid eggs. Eryops is a genus of extinct, primitive amphibians that lived in the swamps during the Permian period, approximately 299 to 251 million years ago. It is an ancient relative of modern amphibians, such as frogs, toads, and salamanders, and bore a closer resemblance to a giant salamander than a toad. This amphibian was substantial and robust, weighing around 200 kilograms or 440 pounds, and measuring between 1.5 to 2 meters, or 4.9 to 6.6 feet in length. Specimens could also reach up to 3 meters or 9.8 feet, featuring a 60 centimeter or 2 feet extended skull and a short, stiff tail. Its limbs were short but robust, with large shoulder and hip girdles. Eryops possessed one of the most heavily ossified skeletons among its relatives, making it one of the largest land animals of its time. Eryops had well-developed ribs, a notable feature considering that most amphibians have minimal or no ribs due to their reliance on cutaneous respiration. This adaptation is particularly significant because modern amphibians, such as salamanders and frogs, do not require ribs for breathing. Evolving from lobe-finned fish during the early Permian, around 295 million years ago, Eryops is considered one of the earliest amphibians. Although it was adapted for terrestrial life, it retained several traits from its Piscean ancestors. For instance, it had lungs for air intake rather than gills, but its ribcage was not fully developed for efficient respiration. Scientists hypothesize that Eryops may have used a pumping motion of its mouth floor to draw in air. The most significant adaptation for terrestrial life in Eryops was its limbs. These limbs, though only marginally strong enough to support its body, were essential for maintaining an upright posture and providing propulsion in water. The hind legs played a crucial role in supporting the animal's weight on land and aiding its movement in water. Walking on land would have been cumbersome and slow, with Eryops likely using short, broad strides. Fossilized footprints suggest that early amphibians, including Eryops, moved in this manner, with more advanced limb structures evolving only tens of millions of years later. Eryops represented a critical stage in evolutionary history, illustrating the gradual transition from aquatic to terrestrial life. As a carnivore, it was one of the dominant terrestrial predators of its era, with a 60 centimeter or 2-foot head armed with sharp teeth, making it an effective hunter. Eryops preyed on fish, small reptiles, and other amphibians, as well as terrestrial invertebrates prevalent at the time. The feeding behavior of Eryops involved waiting with its jaws open, then snapping shut on nearby prey, similar to the feeding strategy of modern alligators and crocodiles. Despite lacking a chewing mechanism, Eryops could clutch and squeeze its prey before moving it further into its mouth. While the exact discoverer of Eryops fossils is unknown, Edward Drinker Cope named the genus in 1887, noting its elongated head. The most commonly found fossils include skull bones and teeth, primarily discovered in lower Permian rocks. Although several complete skeletons have been unearthed, skulls and teeth remain the most prevalent remains. Most Eryops fossils have been found in Archer County, Texas, with additional specimens located in late Carboniferous rocks in New Mexico. Lycaenops is a genus of extinct therapsid reptiles from the Permian period, specifically the Ciceralian era, approximately 290 to 270 million years ago. As a member of the family Gorgonopsidae, Lycaenops offers crucial insights into the evolution of early synapsids. Lycaenops was a predatory therapsid characterized by various distinct morphological features. It had a robust, elongated body and a large skull, which were adapted for its carnivorous lifestyle. The skull of Lycaenops was equipped with prominent saber-like canines, indicating a powerful bite force and specialization in preying upon large, possibly armored prey. The dental structure includes a combination of large, conical teeth and smaller serrated teeth, which were likely used for slicing through flesh. The overall size of Lycaenops was relatively moderate compared to other Gorgonopsids. Adult specimens reached lengths of about 2 to 3 meters, approximately 6.6 to 9.8 feet from snout to tail, with an estimated weight of around 80 to 120 kilograms or 176 to 264 pounds. The limbs of Lycaenops were well developed, suggesting a terrestrial lifestyle with potential capabilities for short bursts of speed and agility. Lycaenops provided valuable information about the evolutionary transition from early synapsids to more derived therapsids. Lycaenops was part of a lineage that exhibited several mammalian traits before the rise of true mammals. These traits include a more advanced dental formula, a more differentiated skull structure, and evidence of a more complex chewing mechanism. Research into Lycaenops and other Gorgonopsids has helped clarify the phylogenetic relationships within therapsids. Studies suggest that Gorgonopsids, like Lycaenops, represent a critical step in the evolutionary history of synapsids, displaying the gradual development of features that would later become prominent in early mammals. Fossils of Lycaenops have been primarily found in the Karoo Basin of South Africa, a region renowned for its rich Permian vertebrate fauna. These fossils include partial skulls, mandibles, and limb bones, which have been instrumental in reconstructing the life appearance and habits of this ancient predator. The fossil record of Lycaenops is relatively well preserved compared to other Gorgonopsids, allowing for detailed anatomical studies and comparisons with other therapsids. The paleoecological context of Lycaenops reveals that it inhabited a semi-arid environment, characterized by seasonal variability and diverse flora and fauna. The presence of Lycaenops in these ecosystems indicated that it occupied a high trophic level as a top predator, feeding on a variety of contemporary herbivorous reptiles and smaller vertebrates. Its adaptations suggest that Lycaenops was an active predator, utilizing ambush and pursuit strategies to capture prey. Edaphosaurus is a genus of extinct edaphosaurid synapsids that lived in what is now North America and Europe around 303.4 to 272.5 million years ago during the late Carboniferous to early Permian. American paleontologist Edward Drinker Cope first described Edaphosaurus in 1882, naming it for the dental pavement on both the upper and lower jaws. Edaphosaurus is notable as one of the earliest large herbivorous amniote tetrapods, four-legged land-dwelling vertebrates. Along with its distinctive large tooth plates, its most recognizable feature is the sail on its back. While other synapsids from the same period, such as the apex predator Dimetrodon, also had dorsal sails, the sail of Edaphosaurus is distinct in both shape and morphology. The first fossils of Edaphosaurus were discovered in the Texas red beds of North America, with additional finds in New Mexico, Oklahoma, West Virginia, Ohio, and fragmentary remains in eastern Germany. Edaphosaurus species ranged from 0.5 to 3.5 meters, or 1.6 to 11.5 feet in length, and weighed over 300 kilograms or 660 pounds. It had a tiny head with shortened cervical vertebrae, massive dorsal vertebrae, a deep tail, and short, robust limbs. Its wide ribcage likely housed a large gut and symbiotic bacteria to aid in the digestion of tough plant materials. Though it had a sail-like structure supported by the vertebral column, Edaphosaurus differed from Dimetrodon by having unique crossbars on the spines that supported its sail. The head of Edaphosaurus was short, relatively broad, triangular in outline, and remarkably small compared to its body size. The deep lower jaw likely had powerful muscles, and the marginal teeth along the front and sides of its jaw had serrated tips, helping Edaphosaurus to crop bite-sized pieces from tough terrestrial plants. Back parts of the roof of the mouth and the inside of the lower jaw held dense batteries of peg-like teeth, forming a broad, crushing and grinding surface on each side above and below. Its jaw movements were propilinal, front to back. Early descriptions suggested that Edaphosaurus fed on invertebrates such as mollusks, which it would have crushed with its tooth plates. However, paleontologists now think that Edaphosaurus ate plants, although tooth-on-tooth wear between its upper and lower tooth plates indicates only limited processing of food compared to other early plant eaters such as Diadectes. The recently described Melanedaphodon from the Middle Pennsylvanian subperiod of the Carboniferous period in North America is currently the earliest known Edaphosaurid and represents a transitional stage from a diet of hard-shelled invertebrates such as insects and mollusks to fibrous plants. The functions of the bony tubercles on the spines remains unclear. It was proposed that the projections on the spines of Edaphosaurus may have been embedded in tissue beneath the skin, potentially serving as food or fat storage, similar to the camel's hump. In contrast, some paleontologists argued that these bony projections were exposed and could have generated air turbulence, thereby enhancing cooling across the surface of the sail for temperature regulation. However, recent studies examining the microscopic bone structures of the tall neural spines in Edaphosaurids have cast doubt on the sail's thermoregulatory role, suggesting that it may have been more likely used for display purposes. A comparative study of bone histology between Edaphosaurus and Dimetrodon revealed that the herbivorous Edaphosaurus exhibited significantly slower growth than the carnivorous Dimetrodon, which showed higher growth rates linked to an elevated metabolism. This slower growth in Edaphosaurus is supported by fewer blood vessels in its bones and the presence of lamellar bone, indicating a lower metabolism and ectothermic traits. Interestingly, despite overall slow growth, the tall dorsal spines of Edaphosaurus suggest rapid seasonal growth of the bony tubercles, contrasting with the incremental development seen in earlier species like Lanthosaurus. Secodontosaurus, cutting-tooth lizard, is an extinct genus of carnivorous synapsids from the family Sphenacodontidae, dating from approximately 285 to 272 million years ago during the early Permian. Similar to well-known Dimetrodon, Secodontosaurus possessed a tall dorsal sail, but it differed in skull morphology. Its skull was long, low, and narrow, with slender jaws and teeth of uniform size and shape, in contrast with the shorter, deeper skull of Dimetrodon, which had prominent canine teeth in the front and smaller slicing teeth toward the back. The unique jaw structure of Secodontosaurus suggests it may have been specialized for catching fish or preying on animals that lived in burrows or crevices. Although no complete skeletons have been found, Secodontosaurus is estimated to have reached lengths of 2 to 2.7 meters, or 7 to 9 feet, and weighed up to 110 kilograms, or 250 pounds. Fossils have been discovered in the Wichita and Clear Fork groups of early Permian formations in Texas. Recent findings by teams from the Houston Museum of Natural Science in North Texas appear to include new specimens of Secodontosaurus, though these have yet to be formally described. The phylogenetic placement of Secodontosaurus among sphenacodontids depends on whether the tall dorsal sail or the deep skull is emphasized as the key trait. In one interpretation, Secodontosaurus groups with Dimetrodon, excluding Sphenacodon. Alternatively, Secodontosaurus could be placed on a distinct branch, while Dimetrodon and Sphenacodon form a separate clade. In one evolutionary scenario, Secodontosaurus, with its elongated, narrow skull, would have evolved from a deep-skulled, tall-sailed common ancestor shared with Dimetrodon. Sphenacodon, which possesses a deep skull but lacks a tall sail, would represent an earlier evolutionary stage, preceding the development of the tall, thin-spined dorsal sail. In an alternative scenario, Dimetrodon, Sphenacodon, and Ctenospondylis are united in a deep-skulled clade that excludes Secodontosaurus. This requires one of two possibilities: either 1, tall, rod-like neural spines, are plesiomorphic in Sphenacontidae, meaning the common ancestor of all 4 genera has a tall sail that was lost through character reversal in Sphenacodon. Or 2, the common ancestor lacked a tall sail, more closely resembling Sphenacodon, and both Secodontosaurus and Dimetrodon independently evolved their tall sails as a derived characteristic. The name Tapinocephalus, meaning humble head, might not sound particularly fearsome, but this creature was a behemoth. Belonging to the group known as Dinocephalians, Tapinocephalus was among the largest land animals of its time, measuring over 3 meters or 10 feet in length and weighing between 1.5 and 2 metric tons, or 3,300 to 4,400 pounds. Despite its imposing size, Tapinocephalus was a herbivore. It thrived in regions that are now part of South Africa, specifically within the famed Tapinocephalus assemblage zone of the Beaufort Group. Fossils found in this area provide invaluable insights into the diversity of Permian life. Although fossils of Tapinocephalus and its relatives are rare, they have given paleontologists a detailed view of this creature's anatomy and lifestyle. Tapinocephalus had a distinct build, short and robust legs, a deep rib cage, and a barrel-shaped body that made it look like a living tank. Its most distinctive feature, however, was its large pachystotic skull. This thickened skull roof wasn't just for show. Many scientists believe Tapinocephalus used it for intraspecific headbutting, similar to the behavior seen in modern-day animals like rams. This headbutting likely played a crucial role in competing for territory or mates. These confrontations would have been powerful but controlled, given their massive weight and size. The heavily reinforced skulls would have acted as a shock absorber, preventing serious injuries during these bouts. Tapinocephalus wasn't just an isolated oddity. It belonged to a larger clade of therapsids known as Dinocephalia. Dinocephalians were an early, diverse group of therapsids, some of which displayed carnivorous tendencies while others, like Tapinocephalus, were strictly herbivorous. This group provides an important glimpse into the evolutionary path that would eventually lead to the rise of mammals. Unfortunately, the Dinocephalians' reign over the Middle Permian would not last forever. The end of the Permian period was marked by one of the largest mass extinctions in Earth's history. Volcanic activity, climate change, and other factors contributed to the downfall of many species, including Tapinocephalus. By the end of the Permian, nearly 90% of all life on Earth would be wiped out, setting the stage for the rise of new groups in the Triassic period. Ophiacodon lived during a crucial time in Earth's history, between the late Carboniferous and early Permian periods, around 310 to 270 million years ago. It roamed the ancient swamps and lowland areas of what is now North America and Europe. With its elongated body and powerful limbs, Ophiacodon was well adapted to a semi-aquatic lifestyle, moving effortlessly between water and land. This has led paleontologists to theorize about its possible ecological role. Was it primarily an aquatic predator, or did it stalk the shores for prey? The name Ophiacodon means snake tooth, a reference to its sharp conical teeth. These teeth suggest it was a carnivore, potentially feeding on fish, amphibians, and other small vertebrates. Its long and narrow skull was unique among early synapsids, giving it an advantage in catching prey in the water or hiding in tight spaces. Ophiacodon possessed a notably large skull with a deep snout, the longest of any early synapsid, with some specimens reaching up to 50 centimeters or 20 inches in length. Its jaws were lined with numerous small teeth, indicating a likely predatory lifestyle. Ophiacodon was larger than most tetrapods of its era, with body lengths ranging from 1.6 to 3 meters or 5.2 to 9.8 feet, and weights between 26 and 230 kilograms or 57 to 507 pounds. Ophiacodon's skeletal structure provides crucial insights into its lifestyle. Its vertebrae and limb bones show signs of rapid growth, with the presence of fibrolamellar bone, a type of tissue that is found in fast-growing, warm-blooded animals like mammals and birds. Was Ophiacodon potentially warm-blooded? Studies of its bone microstructure suggest that it may have had a higher metabolic rate than its cold-blooded contemporaries. This points to the possibility of parallel evolution. Traits that we associate with mammals may have evolved independently in different branches of synapsids. Fossils of Ophiacodon have been found across North America and Europe, particularly in the United States, where its remains have been uncovered in formations across Texas, New Mexico, and Ohio. These discoveries have helped paleontologists piece together its evolutionary history and better understand its role in ancient ecosystems. Pareiasaurus is an extinct genus of Pareiasauromorph reptile from the Permian period. It was a typical member of its family, the Pareiasaurids, which take their name from this genus. Pareiasaurus is a large quadruped, about 2.5 meters or 8.2 feet long, with elephantine legs, walking in a typically reptilian posture. The skull is broad and the snout short. Its skull had several spine and wart-like protrusions. Pareiasaurus's leaf-shaped teeth, ideal for biting through tough plant fibers, indicate it was a herbivore. Even the palate had teeth. Overall, Pareiasaurus represents a key lineage in the history of reptiles, illustrating early adaptations to herbivory and providing important information about the Permian terrestrial ecosystems. Its study contributes to our broader understanding of prehistoric life and the evolutionary history of reptiles. As the Permian period progressed, Pareiasaurus were quite successful in adapting to the changing environment, which is evident from their fossils found around different strata, including the Tropidostoma zone. Their presence in diverse regions underscores their adaptability to various climates and geographies during this era. Their fossils, extensively found across Africa, built a case for a broad ecological niche that the genus occupied during its reign on ancient Earth. Interestingly, a study analyzing reptile phylogeny posits a close relationship between Pareiasaurus and turtles, suggesting that Pareiasaurs might have left a legacy in the form of their shelled descendants. Diadectes is an extinct genus of tetrapods that lived during the late Pennsylvanian to early Permian periods, about 305 to 290 million years ago. This genus belongs to the Diadectidae family, notable for its evolutionary significance in the development of early amniotes. Fossils of Diadectes have primarily been found in North America, especially in the southwestern United States. Diadectes was a large, robust animal, reaching lengths of up to 10 feet or 3 meters and weighing around 500 pounds or 230 kilograms, making it one of the largest land vertebrates of its time. Diadectes is characterized by a mix of primitive and advanced features. Its dentition is particularly noteworthy. It had broad, leaf-shaped teeth adapted for grinding plant material, indicating a herbivorous diet. This marked a significant dietary shift from the carnivorous diets of early amniotes. The robust skull and body structure of Diadectes were well suited for a herbivorous lifestyle. The cranium was broad and flat, designed to handle the mechanical stress of processing tough vegetation. Its stout limbs and barrel-shaped body supported its large size and heavy build. Suminia, an extinct therapsid from the late Permian period, 268 to 252 million years ago, provides critical insights into the evolution of early synapsids, the ancestors of mammals. Described by Vladimir Tatarinov in 1979, Suminia was a small herbivorous reptile about 1 meter or 3.3 feet long. Its fossil evidence found in Russia reveals advanced dental features with differentiated teeth, indicative of a specialized diet. Suminia's anatomy shows a shift towards more mammalian characteristics. Its semi-erect limb posture suggests a transition from the sprawling stance of earlier reptiles to a more upright position, reflecting enhanced locomotion and behavioral adaptation. This shift, coupled with its herbivorous diet, highlights its role in the evolutionary lineage leading to mammals. In essence, Suminia is a crucial genus for understanding the evolutionary developments that bridged the gap between primitive synapsids and early mammals, making it a key focus for paleontologists studying mammalian ancestry. Suminia exhibits a unique combination of dental and jaw features that reveal its herbivorous diet and oral processing mechanisms. Its masticatory architecture, similar to that of Diadectes, is characterized by a sliding jaw articulation. The dentition of Suminia, which lacks canines and features large leaf-shaped teeth, has a completely marginal occlusion. This differs from other species with similar leaf-shaped teeth and indicates a specialized herbivorous diet.